far we have talked about the structure of the spinal cord and its organization. Now we're going to be dealing with the structure of the muscle, which is found on page 208, document A. Now the muscle here contains two kinds of fibers of different function, which are the intrafusal fibers and the extrafusal fibers. Now if you look at the word intrafusal and extrafusal, you would have an idea concerning their location. Intrafusal means in. So if you look here, this is the structure of the muscle here. Okay, as you would observe here, we have the intrafusal muscle fiber. Look at them. They are found in here. They are embedded, that is found within the extrafusal muscle. Here we have extrafusal muscle. These are the muscles here on the outside. Okay, so the muscle is made up of extrafusal muscle here, and embedded within them, inside them, we have the intrafusal muscle fibers. Now, the intrafusal muscle fibers are contain the sensory receptors, which are called the neuromuscular spindle. So if you read here with me, intrafusal muscle fiber are embedded within the extrafusal fiber and contain sensory receptor called neuromuscular spindle, whose central parts are not contractile. Not contractile means that they can't contract. Now I'm going to explain this point here. This is the intrafusal muscle fibers, and here, okay, they took this region and they're showing it to you here. As you would observe, they wrote here above that this is the neuromuscular spindle. So the neuromuscular spindle is contained within the intrafusal fibers. Now, what is the role of the neuromuscular spindle? The neuromuscular spindle is the receptor, the sensory receptor. Remember in our previous session, we clarified that the muscle is a receptor and an effector organ at the same time, because we're going to apply stimulation to the muscle, which is the stretching of the muscle is applied to the muscle itself. We have receptors in the muscle which are sensitive to the stretching. These receptors are the neuromuscular spindle. So the following receptor, the neuromuscular spindle, is found in the intrafusal fibers. As you would look at it closely, you're going to observe that we have the extremity of a sensory fiber, which is going to surround it here. Look at the fiber. Do you see this is the blue fiber, which is a sensory fiber? The extremity, the ends, are surrounding here the fibers, the intrafusal fibers here, and in a spiral way, forming what we call the neuromuscular spindle, which is the sensory fiber. Now, which is the sensory receptor, I mean. Now, we said that this central part are not contractile. They do not contract. Now, the extremity of an alpha motor fiber, here it is written motor fiber alpha, actually it's alpha motor fiber. This alpha motor fiber, which is in red, is going to be connected to the extrafusal muscle, which is forming what we know as the motor end plate, which by now you know that it is the neuromuscular synapse. Now, the intrafusal fiber here also have a motor fiber, which we call it the gamma motor fiber, which is innervating it. Now, what is the role of the gamma motor fiber here? We're going to talk about it later on. Now, you understand what is meant by the muscle is a receptor and an effector organ at the same time. The receptor is the neuromuscular spindle, which is located as I would repeat quickly, in the intrafusal muscle fiber, the neuromuscular spindle is located here, which is surrounded by sensory extremity forming the receptor. At the same time, we have the extrafusal fibers, which have a motor innervation reaching it, an alpha motor fiber. You can observe here you have a clarified figure which is showing you the terminal of the nerve fiber, the terminal end of the alpha motor fiber, the axon, and this is the muscle forming the neuromuscular synapse or the motor end plate. Now, this is the effector. This can be referred to the effector part, the effector organ. So the muscle here is a receptor and an effector organ at the same time. At the same time here too, we have another motor fiber which is going to innervate the muscle. It will innervate the extremity here of the neuromuscular spindle. The following motor fiber are called the gamma motor fiber, and they have a certain role to be discussed later. Finally, we have what is referred to as the Golgi tendon organ. These are localized in the tendon. You remember the tendon from our previous lesson, which are the structures that are going to connect the muscle to the bone. Now, the following tendon, they are sensitive to the tension that is going to affect the muscle, and so we say that they are enhanced to that exerted by the muscle. So for now, this is the structure of the muscle. Now, you know that the muscle has the receptor, which is the neuromuscular spindle. You now know we have the spinal nerve, and we have the roots of the nerve, and we have the spinal cord. Slide 9 is showing here the experiment done to identify the role of the neuromuscular spindle and the Golgi tendon organ. Here, what was done was that they isolated a muscle which is having intact nerve connection. That is, the connection were not damaged. The connection were intact with the spinal cord, and this muscle was subjected to a modification, a change in its length. They used an oscilloscope to record the electric activity of two sensory fibers. I'm going to show you here what records are they, and what do the sensory of nerve fiber where do the sensory nerve fiber come from. Let me start here. We have here the length of the muscle originally, then a weight was attached. As you would observe, the weight is going to stretch it. Okay, so we have here stretched muscle, here resting muscle. Now, as you know that a muscle is going to respond to stretching by contraction, so as you would observe here, the muscle contracted. That is, we agreed previously, when it contracts, it is going to shorten. When it is stretched, it is going to elongate like this. Okay, now what is happening here is they are recording the frequency of the action potential and the sensory nerve originating from the neuromuscular spindle, that is, from the receptor, and the action potential and the sensory fiber coming from the Golgi tendon organ. What was observed is that at first, here, we observe few action potential of same amplitude, like 8, coming from the neuromuscular spindle when the muscle was at rest. While when the muscle was stretched by the mass, the frequency of the action potential increased to 15 action potential of the same amplitude. Now, however, when this muscle contracted, what was observed that in this region here, the region here in the middle, they observed no action potential. Therefore, a conclusion was reached that the neuromuscular spindle is only sensitive to stretching, because we have nervous message recorded when it was in a stretching state, but there was no nervous message here recorded. The neuromuscular spindle is only sensitive to stretching. That is, when it was stretched, it showed an increase in the frequency of action potential. However, when it was contracted, there was no action potential. Now, we go to the third or to the last recording here, where we're going to observe that in contrast here, the sensory neuron coming from the Golgi tendon organ showed no action potential at rest at all. It was not active. However, when the muscle was stretched, in this case, when it was stretched, you observe action potential 7, and when it was contracted, we also observed the action potential 7 also of same amplitude. So therefore, we're going to reach a conclusion here that the Golgi tendon is sensitive to stretching and is sensitive to contraction. We come now to the final slide and the final point, which is how are the following structures, how are the structures that we have explained, how are they all involved in the myotatic reflex. Here we have a schematic interpretation of the myotatic reflex indicating the involved structures. And by the way, this is the answer to probing the activity number 5 in your book. You're asked to draw a schematic interpretation. Now here, let me call your attention to the presence of two pathways. That is, the neuronic circuit here consists of two pathways. An afferent pathway, and by now you know that the meaning of afferent is sensory. So as you would observe here, number 2, centripetal or sensory pathway, which is coming from where? From the muscle itself. Remember, this is the sensory neuron. Its extremity is found attached to the intrafusal, surrounding the intrafusal muscle, intrafusal fiber to form the neuromuscular spindle. So I have here the centripetal or the sensory pathway, which is made up of sensory neuron. I'm going to follow it. Sensory neuron, see the T-shaped neuron, see the cell body found in the spinal ganglion. And this is the spinal nerve, and this is the dorsal root, as you would observe, which is containing the sensory neuron. So this sensory neuron is coming from the muscle itself that is going to be subjected to stimulation here, which is the stretching. We also have an afferent pathway, which by now you know is a motor pathway, which is going to go from the spinal cord to the effector organ. So here, this is the spinal cord. This is the motor fiber. As you would observe, the cell body of the motor neuron is found in the spinal cord, as we have discovered in the previous experiment of Bell and Magendie. And the cell body of the sensory neuron, as I had said, is found in the spinal ganglion. Now, the following sensory neuron is going to go down the ventral root. The ventral root is motor. Here, spinal nerve. This direction is a centrifugal or motor pathway reaching the extrafusal part of the muscle, forming what we call the motor end plate. Now, let me put all of them together. The muscle is going to be stretched. This is the stimulus. The neuromuscular spindle is going to be the receptor, which is going to identify the presence or sense the stimulation, the stretching, generates the action potential, which is going to go along the sensory pathway and the sensory neuron, along the dorsal root, reaching the spinal cord. Now, as you would observe here, we have a synapse between the terminal end here of the sensory neuron and the cell body of the motor neuron. Here, this is a direct synapse. One synapse. This is why we say that we have here a monosynaptic circuit. So, as you would observe here, the terminal end of the sensory neuron is synapsing with the cell body of the motor neuron, and so the connection here between the sensory and the motor neuron are only by one synapse. This is why we say we have a monosynaptic circuit. So the message is going to move along the ventral root, carried by the alpha motor fiber via a centrifugal pathway, reaching finally the extrafusal part of the muscle, which is forming with the fiber here, with the alpha motor fiber, which is forming the motor end plate, causing the muscle to respond to its stretching by contracting. So this schematic interpretation of the myotatic reflex has shown all the involved structures that are allowing this myotatic reflex to happen. I would like you to call your attention to that the monosynaptic circuit requires a very short latent time that allows instantaneous muscular adjustment, meaning that the time needed for the message to go from the sensory to the motor neuron is very short, and this is very important for allowing the muscle to have an instantaneous adjustment, muscular adjustment, to adjust itself to the new situation in which it was placed. This is the end of our session today. You have learned today the structure of the spinal cord and the role of the spinal nerve and the dorsal and ventral root. You have discovered the organization of the muscle in detail and the role of the muscle and the Golgi tendon organ, the role of the neuromuscular spindle, and finally you should be able to make or draw a schematic interpretation as the one found on slide 10, representing the myotatic reflex. Thank you.